Hello everyone, this is Module 7, video number 7, and we're talking about eukaryotic gene regulation. And this video and the next video, those will be the final two on this module. They will be covering different aspects of epigenetics and epigenetic gene regulation. So let's take a look at the outline. And I modified the outline that I posted to abbreviate a little bit some of the topics in this part of the module. And I'll be posting that modified outline as well. So the topic, epigenetic modifications in gene regulation. And I've shown in red what I plan to cover in this video. And the following video will be covering the rest of the part C on this outline. Okay, so I want to talk about, first of all, and kind of giving an overview of what epigenetic modifications are. We discussed this way back in the beginning of the course, so this is kind of a review, and I'll go through this relatively quickly. Then we're going to talk about, what are the actual epigenetic modifications and some features that we didn't talk about earlier in the course. So for DNA methylation and histone modification, what are the features of those that are important in gene regulation? Okay, so let's get right to it. First slide here is describing the history of epigenetics, the idea of epigenetics. And this idea came about from a developmental biologist called C.H. Waddington. He's a British scientist. And he was working on the problem of how in development different cells, starting from a single cell, the fertilized egg, different cells develop that have different properties, different cell types basically. How do different cells differentiate to be able to contract for muscle cells, to send electrical signals for neurons, and etc. How do those differentiate phenotypes come about? So he supposed that since all cells had the same genetic material, there must be something happening above the level of genetics to cause this differentiation, or at least contribute to it. So he coined the term epigenetics. Epi means above. To epigenetics, he defined as some factor that's acting above the level of genetics during cell differentiation in development. I just want to contrast genetics versus epigenetics and point out the main differences between those. So genetics is all about how the DNA sequence, the base sequence, can code for something or how it can be passed on, and it can even be mutated. That's the topic for the last module. So it's all about the DNA base sequence. Epigenetics is about chemical modification of the DNA molecule, but not changing the base sequence. So the epigenetics can modify DNA itself without changing the base sequence, or it can modify the histone proteins around which DNA is wrapped. So, like I said, we have talked about this earlier in the course, and even in the previous video when we talked about the action of activators and repressors, we talked about how co-activators or co-repressors could modify compaction of histones. But we're going to go into a little bit more detail in this video about how they work specifically in gene regulation. Just some definitions related to epigenetics itself. So an epigenetic trait is a trait that is stable through cell division, that results in change in gene expression, but it doesn't change the DNA base sequence. Epigenetics is the study of how these epigenetic modifications come about and how they can change gene expression. The epigenome is referring to the sum of all the epigenetic changes that are occurring in a cell or in a tissue of cells or in an organism. So those are some of the definitions that we'll be using in this video. All right, I want to give you a preview of how epigenetics can change gene expression. And after we're going, this is basically the topic for video number 8. And just to kind of perk your interest in the topic a little bit. So like Waddington suspected, epigenetics does play a role in cell differentiation and multicellular animal development. But it's also been found in another number of other different types of phenomena, some of which are listed on the slide. One is if you have genetically identical individuals, so this could be identical twins, or other types of genetically identical individuals that are produced by, for example, in plants, by asexual reproduction. You can oftentimes tell the difference between those individuals. Think of identical twins. They appear differently, or they may have distinguishing characteristics. Even though they're genetically identical, why are they different and distinguishable? So epigenetic modifications plays a role in that. In some diseases, it's been found that epigenetic modification is involved in passage of traits through alleles from one parent or the other. So this is called genetic imprinting. I won't be talking about this in this module. The process of X chromosome inactivation in mammalian females is an epigenetic process. So we'll go through that in a little bit of detail. And some traits have been found to result from epigenetic modification due to a certain environment in which an organism lives. And these have been connected directly to exposure to toxins in the environment or to variable nutritional conditions, and even variable psychological conditions, psychological trauma, for example. So the phenomenon of epigenetic modification is pretty widespread, and it's a very important, very recent developing area of active investigation. Okay, so the details. How is genetic material epigenetically modified? What exactly is involved? So this is what we've talked about earlier in the course, and this is what I'm going to review pretty quickly here. Two main types of epigenetic modifications can come about. One is direct chemical modification of DNA without changing the base sequence, and the other is chemical modification of the histone proteins around which DNA is wound. Both of these can affect how tightly DNA is condensed in the chromosome. And if you remember, that's called heterochromatin, tightly condensed, or euchromatin, loosely condensed. If DNA is tightly condensed, it's not accessible for transcription, and you consider genes that are packed in tightly condensed DNA in heterochromatin to be silenced. If it's not so tightly condensed, they are accessible, and those genes are on. All right, so the epigenetic code is relating those two types of modifications, methylation to DNA directly, or modification of histone tails. That's going to influence how tightly packed the DNA molecule is. So just a reminder of heterochromatin and euchromatin. The main difference is heterochromatin is very tightly packed or condensed. Genes are therefore silenced, and it stains darker. Heterochromatin is typically higher in AT content. Euchromatin, basically the opposite. Less condensed, genes are on, they're accessible. It has a higher density of genes, which typically correlates with higher GC content, and it stains more lightly on the chromosome. And this is a cytological section looking at the nucleus, and you can see regions of very light staining chromosomes and very dark staining. Light euchromatin, dark heterochromatin. Okay. All right, so methylation of DNA is through directly methylating the cytosine bases in what are called CPGs on the DNA. Whenever a C occurs 5' to a G, this is a target for the enzyme that methylates DNA called DNA methyltransferase, DNMTs. All right? And what gets methylated is the carbon 5 of the cytosine base ring. All right? So that's the methylation site. This is a space-filling model showing that the methylated cytosine, that carbon 5, is located in the major groove of the DNA double helix. Okay? And this is showing that when DNA is methylated and it's not modified by acetylation of histones, which we'll talk about in a second, then this allows the histones to be very tightly packed, and this is how heterochromatin forms. Genes that are tightly packed in histones in the nucleosomes are not available for expression. They're shut off. Okay, one other feature of DNA methylation is it's stable through cell division. And if you remember, in cell division, DNA replication precedes cell division. So if you start with a DNA molecule that's methylated on both strands, and just to go back here, a CPG on one strand necessarily has a CPG on the other strand. So both strands will be methylated. And if you start with that DNA double helix and it gets replicated, remember semi-conservative replication, the parent strand here is going to serve as the template for the formation of the new strand. And DNA polymerases don't incorporate methylated nucleotides. So the new strand, the cytosine, will be unmethylated. However, right after replication, there is an enzyme called DNMT1, which is will immediately methylate the newly incorporated product strand. And so right after S phase replication, there is immediate methylation of the new strand. So methylation is conserved through replication. Right, and this slide is just showing that there are many CPGs typically found in the promoters of genes. Very GC-rich regions, promoters are typically, and lots of CPGs. So all these kind of lollipop type structures are indicating high density of CPGs in promoters. Wherever you find a high density of CPGs, these are referred to as CPG islands. So typically promoters are regions where you find CPG islands. And those are sites for methylation. In a promoter where the CPGs are not methylated, the promoter won't be packed in heterochromatin, and it's accessible for binding of the basal transcription factors. It can be expressed. In a gene where the CPGs are heavily methylated, this will be packaged, compacted into heterochromatin, and therefore not accessible for binding of the basal transcription factors, and this gene will be shut off. Okay? So gene regulation by DNA methylation is going to shut off genes in two ways. One is compacting them into heterochromatin, and the second is by methylation of the promoter, which prevents binding of the basal transcription factors. All right, the other type of epigenetic modification is through is enzymatic modification of histone tails that are protruding from a nucleosome core. All of the histones project these tails, short segments of the protein sequence, and these contain many sites that can be modified enzymatically. So there's a whole host of enzymes that can modify these. There's some that add acetyl groups, some that add methyl groups, phosphate, and numerous other groups as well. Modification of histone tails is going to affect whether they are tightly packed in heterochromatin or not, resistant to packing in heterochromatin. Okay? This slide shows for the 1, 2, 3, 4 histones that are found in the octamer core of the nucleosome. These are the tails, the N-terminal tails that are protruding. And there are various sites. The single letter here indicates the amino acids that are present in location number 5, 12, etc. For each of these histones. We're going to look at mostly at histone H3, and this is indicating that it can be methylated or acetylated or phosphorylated. We're going to ignore phosphorylation and focus on methylation and acetylation. How do those modifications occur, and what are the consequences? All right, so first for acetylation, the enzymes that add acetyl groups to histone H3 tails or other histones is called a histone acetyltransferase, HAT. Okay? So when this enzyme adds an acetyl group to the histone tail, this is going to prevent close packing of nucleosomes. It favors genes being present in euchromatin, and therefore genes being on. Okay? Histone acetylation favors euchromatin formation and maintaining genes in the on status. Acetyl groups can be removed. The enzyme that removes them is called histone deacetylase, HDAC. This is just showing the amino acid that's involved in acetylation. It's called lysine, single letter code K. And histone acetyltransferase is going to add a acetyl group to the end of the R group of lysine, which has an amino group present. Here it is, the acetyl group added to the amino end of the R group on this lysine amino acid. Okay? One thing to notice is that this modification removes a positive charge. Unmodified lysine has a positive charge. Acetylated lysine does not. Okay? For methylation, there's another set of enzymes that are involved. These are called the histone methyltransferases, HMTases. And they can add histone methyl groups to amino acids on histone tails. This can either favor euchromatin or heterochromatin formation. And that depends on which amino acid is methylated. Okay? If you add a methyl group to histone H3, lysine number 9, so that's the 9th amino acid from the end, that favors heterochromatin formation, tight packing of nucleosomes. So this is shown here, methylation of histone tails in neighboring nucleosomes and tight packing that results. This process is also reversible. You can remove a methyl group, and the enzyme that does that is histone demethylase, HDM. And that's shown here. Here is a tail, let's see, and this histone tail can be methylated in different places. If histone H3 is methylated on lysine number 9, that favors heterochromatin formation, and genes would be off. If it methylates a different lysine, number 4, that favors euchromatin formation, and genes would be on. Right? HMT adds methylases, HDM removes methyl groups. All right, this slide is just showing how epigenetic modifications can affect gene expression by either loosely packing or tightly packing nucleosomes. In acetylated histones, there's this loose spacing of the nucleosomes along the DNA strand. And that favors gene expression because promoters and genes are more accessible for basal transcription factors and RNA polymerase II. And methylated histone H3, lysine 9 methylated histones, that favors tight packing, heterochromatin, and not accessible for transcription. Okay, so question comes about, how does methylation result in tight packing? Loose packing is kind of the default state. Acetylation of histones maintains that default state. However, if the acetyl groups are removed and methylation of histone tails occurs, tight packing results. How does that come about? So some of the details about this is known and some of it is kind of hypothesized. What's known is that if histone H3 is methylated on lysine 9, that recruits a protein to bind to this methylated histone tail. And this protein is called heterochromatin protein 1, HP1. And if multiple nucleosomes have HP1 bound to their methylated histone tails, the neighboring HP1 molecules kind of band together into a bundle, and that's called oligomerization. This tightly packs nucleosomes and is what's thought to be the main cause for the tight packing seen in heterochromatin. So this is illustrated on the next slide here. Here is a nucleosome with histone tails that are acetylated, and this is resistant to this HP1 protein binding. Here is a histone with, a nucleosome with histone tails that are methylated, and this fits exactly complementary shape to the HP1 protein, which latches on. So if this happens in a number of neighboring nucleosomes, here they are unmethylated, here they are methylated, here they are with HP1 binding to each of the methylated histones, and here are the HP1 histones all banding together by oligomerization to form the structure in heterochromatin. So the last part is the hypothesized part. That's how it's thought to work. And this slide is just showing what I showed previously, where you can see in a stained nucleus light staining in euchromatin and heavy staining in heterochromatin. All right, this slide is a summary of the epigenetic modifications that we've talked about. DNA methylation, if it's not methylated, that favors decondensed nucleosomes. If it's methylated, that favors condensed, heterochromatin, euchromatin. Histone acetylation favors decondensed nucleosomes, genes are on. Deacetylation favors condensed nucleosomes. Histone methylation can favor either decondensed in some cases, so for lysine 4 on histone H3, or condensed nucleosomes for other locations, histone H3, lysine number 9. Okay, so those are the main types of epigenetic modifications, how they're formed enzymatically, and what are the consequences for gene regulation. In the last video in this module, I'll talk about how epigenetic modification is involved in a number of different phenomena that I mentioned earlier in this video. See ya.